Dear students, in the last session we saw about the cardiotonic diet, and today we are going to look in detail about the role of fat, especially MUFA, PUFA and trans fats. At present, the incidence of non-communicable diseases are increased in most of the countries. The World Health Organization has identified that among the various contributing factors of cardiovascular diseases, diabetes mellitus, improper diet and lifestyle modifications are considered as prime factors responsible. The World Health Organization guidelines in general is to reduce the intake of refined carbohydrate and to replace the saturated fat in diet with mono and polyunsaturated fatty acids and completely restricting the trans fats. The objective of this module is to understand the concept of saturation and categorization of fats and fatty acids and the effects of PUFA, MUFA and trans fat on health. First, we will get to know about the categorization of fatty acids. Fatty acids are the main building blocks of fats. They have a methyl group and carboxyl group with a chain of carbon and hydrogen atoms. About 20 fatty acids are found in our body tissues and in the food we consume. They are categorized based on three aspects such as chain length, saturation and essential fatty acids. In this module, the role of fat is dealt on the basis of saturation and the position of hydrogen atoms. First, saturated fatty acid has a single bond between its carbon atoms, thus the molecule is saturated with hydrogen. Stearic acid is an example for saturated fatty acids. Unsaturated fatty acids have one or more double bonds in their molecule and are thus not saturated with hydrogen. This is further classified based on the number of double bonds as monounsaturated fatty acids and polyunsaturated fatty acids. Monounsaturated fatty acids have only one double bond in the molecules. Example, oleic acid in olive oil. Whereas polyunsaturated fatty acids have two or more double bonds in the molecules, for example, corn oil. Fatty acids are also categorized by the location of their double bonds. The methyl end of the fatty acid molecule is called the omega N. If the first double bond in the fatty acid is in the third carbon atom from the methyl end, it is called omega 3 fatty acid, and if the sixth carbon atom from the methyl end, it is called as omega 6 fatty acid, and when it is in the ninth carbon, it is called omega 9 fatty acid. Linoleic acid, which is omega 6 PUFA, and linolenic acid, which is omega 3 PUFA, are called essential fatty acids because these are not synthesized in the human body, they are not required for important functions and are available only through diet. Trans fatty acids, the hydrogen atoms are in the trans position. Now, we will go into the role of MUFA, PUFA and trans fat in detail. The epidemiological relation between the total fats, fatty acids and human health proves that different fats implicate their effect on health in a different dimension. Each fatty acid has its own biological properties and health benefits. First, the monounsaturated fatty acids. The MUFA are a better replacement for saturated fatty acids than PUFA because they are less susceptible to oxidation. Oxidized LDL is taken up by macrophages and deposited in atherosclerotic plaques. Inhibition of LDL oxidation slows the development of atherosclerosis. It was observed that high intake of PUFA resulted in some lowering of HDL cholesterol along with the levels of total and LDL cholesterol. MUFA is resistant to peroxidation and helps to lower oxidized LDL by preserving HDL levels. More recent studies reveal that high dietary PUFA, that is omega 6, with SFA intake remains the same, may exchange platelet aggregation while MUFA does not. Further, a larger proportion of MUFA in the diet may be conducive to thrombolysis and may be anti-inflammatory. It is now established that substitution of saturated fatty acids by MUFA lowers both total and LDL cholesterol, but not HDL-C. Uncontrolled peroxidation of PUFA in cell membrane may lead to cellular damage, while maintenance of a balance with MUFA is conducive to maintain cell function. Olive oil, extensively used in the Mediterranean region, is rich in monounsaturated oleic acid. Both hypercholesterolemia and coronary heart disease are less prevalent in US, UK, North and Central Europe. This observation led to many studies on the benefits of high fat and high MUFA diet. The step 1 diet and a high MUFA diet were equally effective in lowering total cholesterol and LDL cholesterol without changing the HDL cholesterol. Next is the PUFA. The important omega-3 PUFA are alpha-linolenic acid, eicosapentaenoic acid and docosahexaenoic acid. The omega-6 PUFA are linoleic acid and arachidonic acid. It has several important functions for growth in the young and maintenance of normal healthy skin. They are the components of membranes to ensure their permeability to water and other small molecules. Precursors of eicosanoids, a group of important metabolites which regulate vascular function. The omega-3 fatty acids, docosahexaenoic and eicosapentaenoic acid, have an important role in fetal brain and eye development. These also protect against rheumatoid arthritis and cardiovascular diseases. They are used to synthesize prostaglandins, which prolong bleeding time, reduce the thickness and stickiness of platelets, lowers triglycerides and very low density lipoproteins in the blood. It reduces artery blockage, inflammation and pain in certain ailments. Polyunsaturated fatty acids promote esterification of cholesterol and make into easily utilizable form. PUFA, especially arachidonic acid, are precursors of prostacyclins, thromboxanes and leukotrienes. Thromboxanes are vasoconstrictors, while prostacyclins are vasodilators and help in prevention of thrombus formation. These substances also decrease the aggregation of platelets and decreasing the tendency to clot. Omega-3 fatty acids raise endothelial derived relaxing factors. Hence, PUFA reduces thrombosis. Linoleic acid prevents accumulation of cholesterol in the blood serum and walls of the blood vessels and play a key role in the transport of cholesterol. PUFA decreases the synthesis of the precursor of VLDL, which are associated with an increased incidence of coronary heart disease. Apart from improving the blood lipoprotein profiles, they reduce blood clotting tendency. Alpha-linoleic acid reduces the risk of myocardial infarction and fatal ischemic heart disease among women. This PUFA is further classified into two, omega-6 PUFA and omega-3. Omega-6 PUFA, if carbohydrate is replaced by linoleic acid, the predominant dietary omega-6 PUFA, LDL cholesterol is lowered and HDL cholesterol level is raised. When saturated fatty acids are replaced with PUFA in low fat diet, LDL and HDL cholesterol levels will be lowered. Overall, eliminating SFA is twice as effective in lowering serum cholesterol levels as increasing the PUFA. A 1% increase in omega-6 PUFA would lower total cholesterol by 1.4 milligram per deciliter. However, PUFA have been shown to decrease very low density lipoprotein. Omega-6 PUFAs are widespread in foods, but their major source is vegetable oils, salad dressings and margarine made with oils. An increase in linoleic acid is not advisable above recommended intakes, as larger amounts leads to increase in LDL oxidation. Moving on to the second one, which is omega-3 PUFA. The main omega-3 fatty acids, eicosapentaenoic acid and docosahexaenoic acid, are high in fish oils, fish oil capsules and ocean fish. Most studies have shown that omega-3 fatty acids do not affect total cholesterol. However, they do increase LDL cholesterol and decrease triglycerides. LDL cholesterol tends to be raised in patients with hypertriglyceridemia and lowered or unchanged in normal subjects fed with concentrated source of omega-3 fatty acids. Omega-3 fatty acids lower triglyceride levels by inhibiting VLDL and ApoB100 synthesis and by decreasing postprandial lipemia. The effects of omega-3 fatty acids on triglyceride levels are dose dependent, that is, higher doses produce greater effects. Their greatest clinical utility therefore is with hyperlipoproteinemias, in which the triglyceride level is also elevated. Omega-3 fatty acids, which affect many other steps in atherogenesis, most notably they are precursors of prostaglandins that interfere with blood clotting. Therefore, high intakes prolong the bleeding time. It is now postulated that consumption of fish and fish oils rich in eicosapentaenoic acid and docosahexaenoic acid will lower cholesterol, LDL and triglyceride levels with a subsequent reduction in sudden cardiac death rates. Consumption of as little as one fatty fish meal per week resulted in a 50% decrease in the risk of cardiac arrest after adjustment for all other risk factors. Dietary supplementation with 1 gram of omega-3 PUFA daily reduced the relative risk of cardiovascular death, non-fatal myocardial infarction and non-fatal stroke in patients who had already survived in myocardial infarction. Thus, omega-3 PUFA has cardioprotective effects in primary and secondary prevention. Now, let us look into the important factor, the omega-6 and omega-3 ratio. Humans can desaturate and elongate alpha-linoleic acid into eicosapentaenoic and docosahexaenoic acid, but only when the omega-6 and omega-3 ratio is low. Excess omega-6 fatty acids in the diet saturates the enzymes and prevents conversion of alpha-linolenic acid into longer eicosapentaenoic and docosahexaenoic acid forms. Because the optimal ratio has been estimated to be 2:1 to 3:1, 4 times lower than the current intake, it is recommended that humans consume more omega-3 fatty acids from vegetable and marine sources. Alpha-linoleic acid can be obtained from flaxseed, soybean and green leaves of few plants. Sources of the longer eicosapentaenoic and docosahexaenoic acid omega-3 fatty acids are primarily marine cod liver oil, mackerel, salmon, sardine as well as crab, shrimp and oysters. The FAO and WHO recommend omega-6 and omega-3 fatty acid ratio to be between 4:1 and 10:1. Hope you are all clear about the unsaturated fatty acids. Let us move on to the trans fatty acids. In natural unsaturated fatty acids, the two carbons participating in a double bond each bind in a hydrogen on the same side of the bond. Here, the hydrogen gets attached in the trans position with one hydrogen on opposite sides of the double bond. Trans fatty acids raise LDL cholesterol to the same extent as myrcetic acid and in addition lower the HDL cholesterol. Thus, trans fatty acids turning out to be even more atherogenic than myrcetic acid. Further, trans fatty acids have been found to raise lipoprotein levels, thus raising the risk of coronary heart disease. 50% of trans fatty acid intake comes from animal foods and the other 50% comes from hydrogenated vegetable oils. Hydrogenation of vegetable oils alter the geometric structure of the PUFA from natural cis to trans forms. Only cis forms can serve as essential fatty acids to be incorporated in cell structure. Depending on the degree of hydrogenation, trans fats in food products contain anywhere from 5% to as high as 40%. Ellaric acid, the trans isomer of oleic acid, raises the blood cholesterol when compared to PUFA. However, it has less of a cholesterol raising effect than the saturated myrcetic and lauric acid. Therefore, consuming trans fatty acids at level 3% of kilocalories will raise the LDL cholesterol levels, but to a lesser extent than saturated fatty acids such as butter. Increased trans fatty acid intake also lowers HDL cholesterol. Consuming approximately homemade low fat foods, low fat dairy products and low fat meat will lower trans fatty acid intakes. In the process of digestion, the enzyme lipase aids in the metabolism of fat, but only in the cis form, although this theory has been modified by the upcoming researchers that the trans fats are metabolized, but it competitively inhibits the ability to metabolize essential fatty acids including omega-3 PUFA, leading to changes in the phospholipid fatty acid proportion in the aorta of the heart, thereby increasing the risk of coronary heart disease. Trans fat are metabolized by the liver with a different approach than the other fat and interferes with the delta 6 desaturase. This is an enzyme involved in the conversion of essential fatty acids to arachidonic acid and prostaglandins, which are important factors for proper cell function. To summarize, reduced intake of refined carbohydrate, replacing the saturated fats in the diet with mono and polyunsaturated fatty acids, complete restriction of trans fats helps in protecting the cardiovascular health. Hope you all understood the topic and thank you for listening. Let us see in the next session.